I think that should work. All right. So, um, hi. Hello, everyone. So my name is Irina Kopyeva, and I am a second-year PhD student in the DeForest Lab in the Department of Bioengineering. Today, I'll be talking about cells and gels, or how we can better design biomaterials to study cells and their environments. So when we think of studying cells, we might picture scientists in the tissue culture hood with flasks and petri dishes and growing cells subsequently on the bottom of these. And so these cells end up growing in a flat layer. This has obviously been vital for a lot of discoveries. But let's take a step back and consider the human body. We are inherently not two-dimensional. We're three-dimensional. Following that path of logic, our cells grow in a 3D environment. So when we study cells on a two-dimensional flat plate, we might be losing some information. So what actually surrounds cells? What makes up the space between them? So if we take our biology textbook image of a cell and zoom in right outside of its membrane, we encounter a really dense mesh. This mesh is primarily made up of sugars and a bunch of cell-secreted proteins that are all intertwined. This is known as the extracellular matrix, or as I'll refer to it, the ECM. The ECM can be thought of as a scaffolding for the cells, and as such gives tissues and organs their shape. The ECM has been found to have many roles in cell function, development, movement, and so forth. These contributions can be divided into three domains. So biochemical, the ECM traps a lot of signaling molecules and proteins, which influences what cells interact with. The topographical contribution, so the arrangement of physical features actually has been shown to influence cell organization and even stem cell fate. And then finally, mechanical contributions, which I'm mostly focused on. So if we look at our bodies, we can see that our cells are in a variety of environments, which massively influences their function. For instance, fat and brain cells find themselves in a really soft environment, whereas cartilage and bone cells are in a really stiff environment. Moreover, this is constantly changing during the cell's development. It's interesting to note that the plastic we culture cells on is relatively similar to the stiffness of bone. Furthermore, the stiffness of the ECM also drastically changes in many diseases, one of the most striking being breast cancer. So in the doctor's office, often breast tumors are detected by physically feeling for a stiffer mass. And it's been demonstrated that the stiffer environment breast cells find themselves, the more malignant they become. So to study these diseases, we should also really be considering their environment. So in order to model the ECM, one of the methods researchers have proposed has been to utilize polymers to replicate the network that is the ECM. We can design polymers, or really long molecules with repeat chemical units, that have similar properties and are interconnected by specific peptides. And when the peptides react with the polymer, they too form a water-swollen network that's very similar to the ECM. These water-swollen polymeric networks are known as hydrogels. Hydrogels are actually everywhere, from contacts to baby diapers and to even bubble tea. These polymers have been shown to be biocompatible and even support cell growth. So as such, my goal is to develop a hydrogel system which can change stiffnesses in real time so we can track cells' response to these changes and hopefully utilize this knowledge from a 3D system to better understand tissue and disease development. To do this, I have been developing a system using two polymeric networks that are created by different chemistries. Each of these systems independently offers some mechanical contributions, but when we add them together, they create an even stiffer environment than the sums of their individual contributions. This is due to their interwoven nature. So if you think about a net tangled in another net, that would create kind of a stronger overall mesh. So true to the idea, when I've measured the mechanical contributions in this graph, which is denoted by the storage modulus on the y-axis, or in simpler terms, the stiffness, we can see that the double network has a higher storage modulus than the sum of the two individual networks. Additionally, these two networks can be degraded independently on demand due to the design of the peptides. For instance, I can expose my first network to light or an enzyme, which is a protein in our case that cleaves in a specific area, and the peptide making up the bond between the polymers is cleaved. Similarly, the second network can also be degraded by another input to release everything. I've made a few variations of double networks, but my first design involves using polymer networks that degrade in response to two different enzymes. We can see in this figure that the gel forms and it makes this really stiff environment about 6,000, 7,000 pascals. And then I add one enzyme in and one network starts degrading. And then when I add in the other enzyme, the other network degrades as well. Similarly, I've also done it with a light-based approach. When we shine light, we see the first network rapidly degrade, and then when we add the enzyme, the second network also degrades. We can also readily tune how much the first network degrades, and hence the stiffness, by changing the light exposure. Ideally, once I finish optimizing the system and can obtain the exact stiffnesses, I'll be able to move towards studying cells and their response. For instance, we can activate the cells in the stiff gel, then drop the stiffness, and finally, we can then fully release them for downstream studies, such as how that affected their gene expression or transcription. So far, I've been able to demonstrate that these double networks are amenable to cell survival, and now I'm currently moving forward with testing specific cell type activation and response, but unfortunately, I don't have any cool photos to show you guys just yet, so stay tuned. So thank you so much for listening to my talk, and I hope you learned something new. Here's my contact info.